Bio 30. Okay, so this is the first lecture on the last unit that we're going to discuss. This is actually worth 40% of your mark in Bio 30, so there's quite a bit of information that we'll be discussing over the next 6-7 weeks or so. So we have a lot of time to do this unit. In your key, cell division, genetics, and molecular biology starts on page 73 and then ends on page 117. I'm not really going to address much in the key today. I'm just going to give you an overview, and I'll talk more about mitosis and meiosis in the next few days. But as we get started, first thing that we need to review is review some stuff from Science 10. So we're going to go back to surface area to volume ratio, and we're going to talk about why cells are small. Cells are small because transportation between selectively permeable membranes are most efficient when we have a high surface area to volume ratio. So when the outside or the borders of the cell is a very close ratio to the volume itself. You can think about this diagram right here. So you can see, so let's say this is a cell, and then this is the nucleus of these different cells. So this might be a little bit of a deceiving photo, but all of those purple balls in the middle of these squares, they are all the same size. So you can see that if the cell itself is small in comparison to the rest of its contents, then it doesn't take as much distance for waste to be diffused out of a cell and nutrients to come in. So cells have to be small in order for efficiency of transport to be the greatest. Because if you see this big cell, you can see that if the surface area to volume ratio was small, so a really large volume in comparison to the borders of the cell, it would take a long time for transportation to occur. So this is why when we look at cells of an elephant and a human and a dog and a small animal like an ant, their cells are all kind of the same size. The big difference is that bigger animals just have more cells. And that's because efficiency of transport is optimized when we have a maximal amount of surface area to volume ratio. So just remember that when we're talking about mitosis and meiosis and these other cellular processes, that we're talking about microscopic divisions. And it's because cells have to be small in order for that to happen. Okay. So the cell cycle is, so we're going to talk about mitosis and meiosis, right? And you learn in grade 9 about mitosis. Mitosis is the division of cells for growth, maintenance, and repair. Meiosis makes gametes or sex cells. So we're going to talk in general about these two different modes of replication. Mitosis has one division and makes two daughter cells, and that happens in body cells or somatic cells. Meiosis happens in gametes or gonads, so sex cells, and the purpose is to make sex cells, so sperm and egg, and they're haploid rather than diploid. So I'm going to talk about what that means in a second. Okay. All right, so in somatic cells, the genetic material of the original parent cell is duplicated, and we make daughter cells that are identical. In meiosis, we make 4 daughter cells that are genetically distinct. So we'll talk about that when we get to meiosis. But the big thing that we should understand with the cell cycle in biology is, I'm going to actually put this on my board. Okay, so this says M phase. So when we look at the life cycle of a cell, there's basically 4 different distinctions. G stands for growth. So G1 phase, S phase is called synthesis. G2 is another growth phase. So the cell actually dividing in the M phase is actually a very small part of the cell cycle, right? So if you think about, right, the cell has to grow and divide and live its life out. So the cell kind of lives this through here, through the G1, S phase, and G2 phase. It doesn't spend most of its life dividing. It actually just spends most of its life living. So we have the M phase, and mitosis is the division, and we have our different stages of mitosis that happen there. And the rest of this is interphase. So the G1, S phase, and G2 phase all happen in interphase. And the rest of the actual division of the cells, when they're ready to divide, happen in the M phase. And then you have prophase, metaphase, anaphase, and telophase. So when we look at the cell cycle, we use this acronym IPMAT, interphase, prophase, metaphase, anaphase, and telophase. But the cell actually spends most of its time in interphase. So that's a significant piece of information for you to understand. So when we look at what happens in interphase, G1 is when the cell is newly divided, the cytoplasm expands, the cell matures into an adult cell and carries out the metabolic processes that it needs to do. S phase is where DNA is replicated. This is going to become very significant as we talk about mitosis and meiosis. So in order to divide, the DNA needs to be doubled, and this is what happens in S phase. G2 phase is cytoplasm expansion, and the cell is getting ready to divide. So it's kind of puffing itself out in order to be able to divide into two cells. And then obviously that M phase is the division, all right? So that's the basic idea of the cell cycle. Okay, I'm not going to talk much about the stages of mitosis today. I'm just going to give you an introduction about terminology. Right, so this is what happens here. Right, so mitosis is the division of the genetic material and the contents of the nucleus of somatic cells into two complete and separate sets. So you should be aware of that. Okay, and my notes just kind of explain these phases that I just put up on my chalkboard wall. Okay, so again, all these are in your notes. Remember, it's under Cummings' notes. This will be under cell division and genetics. We're talking primarily about cell division in eukaryotic cells, right? We're eukaryotes. A eukaryote is something that has many different organelles. So eukaryotes have membrane-bound organelles. So again, like things like the nucleus and the Golgi apparatus and the so all those different little organelles, eukaryotic cells have. Prokaryotes do not have that, right? They only have cytoplasm and DNA extended through the cytoplasm. So the cytoplasm has to kind of do everything. So in eukaryotic cells, in prokaryotic cells, but it's a bit more complex in eukaryotic cells, we have DNA, and that's the genetic material, right? So the genetic material are the instructions to build organisms, right? So we have a genetic code that determines what your structures look like and your behavioral characteristics look like in tandem with how it interacts with the environment, right? So DNA is actually a complex. It exists inside proteins called chromosomes, right? And the DNA itself is a nucleic acid that. So chromosome is a combination of DNA and proteins called histones. All right. So they compact DNA. Histones are responsible for the expression of different genes. So you have, if you look at DNA material, most of the time it kind of exists as this squiggly, uncondensed stuff, right? It's double-stranded, right? But if you have alleles, right, you could have two copies of alleles, which code for the same trait. What happens is these histones, they ravel and unravel DNA. right? So the DNA is raveled up here, and then the DNA is unraveled. Okay, so histones kind of determine what alleles are expressed as a result, so forms of genes, and which ones are not expressed. So if the histones have a set of characteristics on a DNA strand sort of wrapped up, the replication machinery can't get into there and we don't actually express that trait, but you're always going to have one copy of the trait expressed. So you always have two copies of each trait. And how the histones are packaged is largely determined by the environment. So your interactions and behaviors with the environment determine which alleles get expressed. Some of it is random, but some of it has to do with the environment. So that's called epigenetics, right? So we call that above the genetics. So it's not only what you get inherited, but it's how you interact with your environment, which determines which set of alleles on the DNA gets expressed. So, right, so that'll give us different behavioral characteristics. So it's a really important field of science. So we're not going to touch a lot in this unit of genetics, but remember that epigenetics is just as, if not more important than understanding the DNA structure itself. So how the structure is expressed is quite important. So histones help determine the structure of DNA. You'll also have on the ends of DNA strands, you'll have these repeating units called telomeres. So DNA is composed of 4 nitrogen bases. So you have adenine, cytosine, thymine, and guanine. Okay, now we're going to talk about that more, but all genetic code on Earth uses these 4 base pairs, and it's the order and the amount of those base pairs that determine different species and differences between organisms within a species, right? So we have, for humans, we share 99.99% of the same genetics, but we have a few different base pair codes that have different orders, and that's what gives us all of our different traits and characteristics. But every organism on Earth uses the exact same base code. So that's obviously shows that that's common ancestry, right? So these telomeres or telomeres, they're repeating units of adenines and thymines. And these, they're kind of like drawstrings on the edges of DNA. They don't actually code for any traits, but what happens is every time these divide, the telomeres get smaller. All right, so you kind of cut off parts of the telomeres. With each division. All right, so these extra drawstrings, when the cells divide, keep the base pairs that are coding for traits, from being messed with or mutated and or removed. So, but the thing is as divisions happen, these telomeres get smaller and smaller and smaller, and eventually they're gone, and this destabilizes the DNA molecule. So telomeres are a big thing that causes aging, that and the accumulation of mutations. So, there's, whether it's ethical or not, there's lots of scientists working on solving the aging problem, and they're working on how do we keep these telomeres themselves from getting smaller and smaller and smaller, and then destabilizing the DNA molecule as those telomeres get smaller. Okay. Oops. So for most of DNA's life, DNA exists in the nucleus of the cell, so it never leaves the nucleus, it's always there. So think of the DNA, it's kind of like a filing cabinet, so the DNA is always in that filing cabinet. You can remove copies of files, but you can't take the filing cabinet out of the room, right? So we can photocopy copies of files that are in the filing cabinet, but we can never remove that filing cabinet from a room. And that's kind of how DNA works. So we can take the instructions out and use it, but we can't take them, we can take the instructions out and remove them by copying them, but we can't take the DNA out itself. All right, so for most of the time, DNA exists in the nucleus of a cell is this uncondensed material called chromatin. When the cell is ready to divide, it turns into chromosomes. So what happens is the histones contract and make this chromatin a lot closer together, and you end up with this double helix structure. Okay, and then we'll end up with these chromosomes with a little piece in the middle called a centromere. Right, so this chromosome or this piece of DNA material will have, a histone protein in the middle, and that's called a centromere. So the histones contract and bring all that DNA together. It's kind of like having twine on a bale of hay or something like that. Okay. So you guys have seen DNA like this before, whether you kind of studied it closely or not, but it's not actually how DNA exists most of the time. It's kind of this squiggly mess of uncondensed material. Okay, so there's some numbers that you should understand. So humans have, under normal conditions, humans have 46 total chromosomes, but they are in pairs. Okay, so you get one set of genetic information from maternal origin or from your mother, and one set of genetic information of paternal origin or from your father. And you have to have copies of the same trait. So you have two copies of each trait, and they either come from mom or from dad. And so DNA exists in pairs. So you have 46 total chromosomes, or 23 sets. So human somatic cells or human body cells will have 22 what we call autosomal chromosomes, so chromosomes that don't code for sex organs, and one set of sex chromosomes, and that's the sex determining chromosome, so the XY or the XX. So we have 23 sets of chromosomes, and that leads to the term ploidy. So a diploid cell, so if we think about what this means. So we have 23 sets of chromosomes. And these chromosomes are called homologous pairs, meaning that they code for the same traits, but they have different versions of those traits. So they have different alleles because you're getting them from either your mother or your father. But you have to have two sets of each trait, like hair color or eye color, or different types of personality or protein structures that build organs in your body. You have to have two copies of those in order to be a fully functional human being. So we use this number 23. So n equals 23 because we have two pairs. So because we have two, right? Because we have two sets of each, 23 times 2 equals 46 chromosomes. So humans are 2n. So we're 2n because we have two copies of each chromosome, and that's called diploid. Okay. All right, that's called diploid. And you could have, and humans also have haploid cells. And that n number is 23. So n equals 23. So 2 times 23 is 46. So you are all diploid organisms. But we also have haploid cells as well. And those are your sex cells, because the goal is that you get one set of 23 chromosomes that's going to combine with another set of 23 chromosomes to make, 23 pairs and 46 total, which makes a new person when fertilization happens. Okay, so that's ploidy, so you should understand that. Okay. So these pairs of chromosomes are called homologous pairs. And homologous pairs, what they do is they carry the same genes, right? So they have alleles at the same gene locus or loci, so the location of where the genes are. So they're going to be in the same place. So they have the same genes, they code for the same types of things, but they have different forms of those traits. All right, so one's going to be maternal and one's going to be paternal. They code for the same things, but they're not necessarily going to be the same form of that thing. So they carry different alleles. So form of a gene is called an allele. All right. So this is a fundamental concept. This is actually probably the most important concept that you're going to need to know is what is a homologous pair. Okay. So we have diploid and haploid cells. Right, our gametes are haploid in hopes that a gamete fertilizes another gamete and makes a new diploid organism. But you don't necessarily just have to have haploid and diploid. So there's lots of animals that have polyploidy, so lots of plants do. So this is a big way that we genetically modify organisms, by the way, is we induce what we call non-disjunction, so uneven chromosome splitting during gamete formation. And those cells end up having different ratios of alleles or chromosome sets, and that does things like for example, strawberries will induce non-disjunction through like ultraviolet irradiation, and that will make chromosomes split unevenly, and then they'll be, rather than being diploid, they might be triploid, and that extra set of chromosomes will make strawberries grow bigger, for example. All right, so you don't, I'm going to talk about the salamander here in a second, but you don't necessarily need to just have haploid and diploid. So anything that's not haploid or diploid, so above haploid or diploid, is called polyploid. So you could have triploid cells. You could have, this is tetraploid. You could have anything that's an even number that you can split, right? This is pentaploid. Right, so when we look at humans do this. So humans have one sex cell, so sperm plus egg. They will fertilize to make a diploid zygote. Right, and then when cells divide in meiosis, we actually make 4 haploid gametes. Okay, but in some organisms, this is what happens. So let's say we have 2N. We replicate the DNA and divide the cells. Sometimes there's uneven gametes. And then let's say we have another organism like this. And then let's say these two, so these are gametes. Okay, so these are gametes. And let's say this is a theoretical organism. You could have these gametes combined to make a tetraploid organism. Or you could have these gametes combined and make triploid. Or you could have these gametes combined and make diploid. So there's different combinations that could happen based on the environmental conditions at hand. One thing that has to happen with the division of gametes is they have to divide in even numbers. You can't have like 1.5 gametes, for example. But different combinations can arise. This happens in plants quite a bit. A good example of this are the tiger salamanders. I think I discussed this either in Bio 30 or Science 30 a few weeks ago before school was out. We have tiger salamanders in Alberta. Now, tiger salamanders are usually diploid, but in some cases, you can actually have a diploid gamete and a haploid gamete come together to make a triploid organism. Okay. So this is a tiger salamander. You may have seen this around. Now, in the fall, sometimes you see these really massive salamanders, these big orange-looking guys. Let me see if I can get a picture of one here. Just give me a second. Okay, so this came from the exact same. This is the exact same species that I just showed you on my slides, but this is what we call a berserker male. It's kind of like some weird ass Warcraft terminology, but this is a berserker male tiger salamander. So if you guys have seen these, you see them on occasion on the trails around here. This is actually a triploid salamander. So it's a male, but sometimes the gametes combine to make this triploid dude. So in the fall when they reproduce, sometimes the weather turns for the worse really quickly, or there could be drought or early snow. If the regular males haven't had time to mate, some of these triploid guys, they'll actually turn from being that nice, calm, regular-looking tiger salamander. They'll actually morph into this. So it's a reaction to testosterone. And they end up being these really large, aggressive animals. And what happens is that it's kind of like what happened to, I guess, if you remember watching Wolverine, the last Wolverine, right? He takes the serum and then ends up being invincible for a short period of time and then just falls apart, right? But that's what happens with this guy. So this triploid salamander has this extra chromosome, and it allows some genetic mutations for him to increase size. And if it's a scarce year where there hasn't been a lot of matings, these guys can sort of bash through the bush and find a female and mate. They expend their resources really quickly, and they'll end up dying or being preyed upon, but it will allow them to reproduce. So there's this unique reproductive strategy that these salamanders have with messing with their ploidy. Another great example of ploidy in Alberta, or different types of ploidy in Alberta, are trout. So stocked trout in Alberta, if you go to a put and take fishery, not in a wild river, but in a stocked lake like Grotto Pond or Chain Lakes, anything like that. Anything where the province stocks fish where you can take them, like Mitford Pond, you can take them and eat them. Those are all triploid organisms. So they've had triploidy. So they're 3N. They've had triploidy induced into them. So if they get into a wild environment, they can't reproduce. So we want wild stocks to reproduce naturally. We don't want to artificially induce that. So that's another example of where ploidy is unique. So ploidy is your chromosomal complement. All right. So it's the mathematical ratios of chromosomes that are present. So in humans, we're diploid because we have 2 copies of each gene. A triploid organism would have 3 copies of each gene. And in rare cases in humans, we do have triploidy, and I'm going to get to that. So here's how we determine this. So we actually look at, we can extract DNA, and we can move it through a machine called an electrophoresis machine. I'm actually going to talk about that at the last part of the unit. But we can cut pieces of DNA up with enzymes called restriction enzymes. We're going to talk about those when I talk about genetic engineering. And then we can put them in an electrophoresis machine and then separate them. And when we've separated them, we can get a display or a set. And a display of chromosomes is called a karyotype. So this is a human karyotype. So if someone's going to go to the doctor, if they're expecting, and they have an embryo or a fetus, and they want to know if there's any genetic abnormalities, they can either get an amniocentesis or do chorionic villus sampling to sample the kid's DNA. So this is a human karyotype. You can see that there are 23 sets of chromosomes. Chromosome set 23 is this, XY, so this is a male. And you can see that the chromosomes are separated by length, shape, and where the centromere is. So the centromeres are all in different places, so the middle part. So these are all genes that have the same set of alleles. So they're the same genes, but they have different sets of alleles. So they all code So chromosome 10 codes for the same traits, but you may not get the same form of each trait from your parent, and chromosome pair 1 wouldn't code for the same traits as chromosome pair 10, and and likewise with all the other all the other chromosomes. So if I look at a chromosome complement, if this is chromosome pair 1, there would be a chromosome from mom and a chromosome from dad. And then during the S phase of interphase, we would replicate each, we'd replicate the DNA from mom and replicate the DNA from dad. So this would be a chromosome that has a duplicate. Okay. And this would be your maternal chromosome and your paternal chromosome. And they've now duplicated, so we can split them to make two new cells for mitosis. But this set of chromosomes is called a homologous pair. So it's a set of chromosomes that has the same genes, but codes for different alleles. And this is also a homologous pair, but it has its chromosomes duplicated, so there's an extra set of DNA there. I'm not going to discuss that today, but that is a homologous pair. So when you look at karyotypes, you have 23 homologous pairs of chromosomes. Okay, so one comes from maternal origin, one comes from paternal origin. Now you need to be able to recognize some different types of karyotypes. So this is a normal human male, but we can look at some different karyotypes that show different situations. Okay, so if we look at, for example, an individual who may have been born with Down syndrome. This is, if I can pull up a larger version of it. Okay, so here's a good example of Down syndrome. So you can see that there are 23 chromosomes, and so the X, so you know that this is a boy, because the X chromosome is much larger than the Y. So this is a Y chromosome. It's very, very small. So this determines whether or not testosterone is present. So this will turn the female erectile tissue into a penis, and that's an X chromosome. Okay. So with Down syndrome, there's actually not 46 chromosomes. There's 47. And if you see here, that's chromosome pair 21. There's actually an extra chromosome there. So there's been non-disjunction or uneven chromosome splitting. So an individual